Right, ladies and gentlemen, I think we've a very, very good audience here this evening. And I would like you to welcome you to the Ulster Beekeepers winter webinars. Now, normally at this time of the year, we normally have a conference in which we will invite some international speakers, and it is usually a very, very successful weekend. However, unfortunately, due to COVID, we have been unable to put on our normal conference. So instead, we've been running a series of 6 webinars, winter webinars, through January and February and March, or January and February. My apologies. And indeed, the first 3 have gone extremely well, and I'm delighted this evening that we have Professor Juliana Rangel from Texas A&M University to give us a lecture this evening on Queen Management Essentials. Now, for those of you who don't know where Texas A&M University is, it is near to a town called Bryan, and it's about 100 miles northwest of Houston in Texas. A little bit of, housekeeping. You have indeed some buttons at the bottom of your screen. There is a chat button, and certainly we're delighted if you wish to just send a greetings into that particular chat room. Beside that, there's a Q&A button, and then please, if you have questions to ask the speaker, could you please put them into the Q&A. And I'm delighted this evening to welcome Professor Juliana Rangel. Juliana was born in Texas, and excuse me just a second.
In Colombia.
She was born in Colombia.
Colombia, South America.
She obtained her Bachelor of Science degree in 2004 in Ecology, Behavior, and Evolution from the University of California in San Diego. And in 2010, she obtained her PhD from the Department of Neurobiology and Behavior at Cornell University in Ithaca, New York, working under the supervision of Professor Tom Seeley. Professor Seeley, who many of you will know, is probably one of the greatest bee scientists on the planet, and he has a very high regard for Juliana's work. In 2010 to 2013, she worked in North Carolina with Professor David Tarpey. And then in 2013, she joined Texas A&M University and is now the Associate Professor in Entomology. Her research program focuses on the biological and environmental factors that influence the reproductive quality of honeybees, queens, and drones, the health and population genetics of feral honeybees, and the quality and diversity of floral sources collected by honeybees in developed areas around the country. She is an active member of Texas Beekeepers Association and has spoken to numerous beekeeping associations in Texas. And I understand that the drones in Texas fly around with large hats, cowboy
hats. And cowboy boots.
She is a co-advisor of the Undergraduate Entomology Student Organization, and in 2014 was president of the American Association of Professional Apiculturists. So, ladies and gentlemen, it gives me great pleasure to welcome Professor Juliana Rangel to give us our talk on Queen Management Essentials. And this lecture is sponsored by Maysmore Apiaries, and we thank them very much for the sponsorship. Juliana.
Hello, everyone, and thank you so much for the invitation. I am going to share my screen before I get started. So first I need to ask if you guys see my screen. And I don't, they raised their hand, so that means yes.
I'm seeing it, yeah.
Okay, good. So Again, I want to thank you all for inviting me. Me and my family are indebted to John and Susie Hill for inviting us over to Northern Ireland a couple of years ago. It's still one of the great times we had, traveling internationally, and I hope it's not the last. Your country is beautiful, and so unfortunately we have to do this virtually, but I'm still delighted to be speaking to you. So thank you for the invitation to speak to your group again, and to your sponsoring organization for sponsoring this talk. So today we're going to talk about queen management essentials, and it's not really heavily or a research heavy talk, but it's going to go over the factors that influence the reproductive quality of honeybee queens. And so, okay. So when we think about honeybee queen quality, we have to think about several factors that affect their health, including genetics, mating conditions, age and vigor of the queen, pesticide exposure, pathogen loads, parasites, in particular, of course, Varroa destructor mites, the quality of nutrition or lack thereof, habitat fragmentation, climate change, and the most important one, I think, that we sometimes don't pay attention to is our own beekeeper practices. So I'm going to go over each one of these, and I'm going to give you some examples of research that has been done in these aspects of the queen management essentials, including some of our own work in my laboratory. So first, we're going to start with genetics and why the queen's genetics and the genetics of her drone partners are very important to keep a good queen. So first, we know that queen quality depends a lot on her strain or race, and that comes from the genetic diversity of her parents, the type of breeding paradigm that she has been brought up from, and the degree of inbreeding that she may have, and hopefully she won't have much inbreeding at all, but it happens a lot, especially when there is no new stock being brought in from outside where we do queen rearing. So in particular, genetic diversity avoids inbreeding problems because inbreeding can cause production, in particular, of diploid drones. So I want to bring to your attention what the production of diploid drones entails. Honeybees are not only haplodiploid in that queens and workers develop from fertilized eggs that are diploid, having two sets of chromosomes, one from the dad and one from the mom, and drones develop as haploid individuals, having one set of chromosomes only from the mother. But they also have a very interesting sex-determining gene that is unique to honeybees. So in honeybees, sex is determined by one sex-determining gene that has 15 different alleles or variants, or let's call them flavors. So if an egg is unfertilized, that individual has only one allele at the sex-determining locus, and the bee turns into a drone, a normal drone, and that's what we know about haploid drones. But if the egg is fertilized, that individual has two alleles at that sex-determining locus, and so there's two possibilities. If the alleles are different, the bee turns into a diploid female, and those are the vast majority of diploid organisms in the colony. But if the alleles are the same, the bee turns into a diploid drone. Perhaps you've heard this before. You guys are very well informed beekeepers in Northern Ireland, but I will explain this a little bit further. So as we know, queens are diploid. In this example, this queen has two alleles at the sex-determining locus. She has allele A, or call it green, or circle, or whatever, and the other allele that she got from her other parent is allele B. Then she mates with a series of drones, in this case, 5 different drones, but because she happens to be a little inbred, she's mated with drones that have that happen to share one of her two alleles at the sex-determining locus. In this case, allele A for this drone, and in this case for drones 2 and 4, allele B, which she also has. And so in the case of her offspring coming from this drone that has the allele A at the sex-determining locus, there's two homozygous alleles here for this particular diploid individual. So the same AA, the same for this one here would be the queen has the allele B and the drone has allele B. So this individual here is homozygous at the sex determining locus, the same for this one here. For the other drones that do not share any of the alleles of the queen at the sex determining locus, it doesn't matter. It can be going from C to H or I, however many sex determining alleles, how many alleles there are, in this case 15, then all of the offspring are going to be heterozygous at the sex determining locus, meaning that they will be different and therefore the bee will turn into a diploid female. But for these highlighted examples, those individuals are going to be diploid drones. And so that inbreeding causes a higher likelihood or a chance of diploid bees having the same alleles at that sex determining locus, and those diploid drones are not viable. The workers actually eat them before they emerge, typically. Too many diploid drones cause what is known as shock brood pattern. And so you may have a colony where it looks like the queen may have a decent or used to have a decent egg laying pattern, but you see a lot of hole or openings and a lot of empty cells. And it may be because some of those diploid organisms were not going to be developing as workers, but rather as diploid drones. And they get typically removed during the pupal stage. If you've seen pictures of adult diploid drones that may have emerged from cells before getting cannibalized by the workers, they look almost albino and they're not viable drones, so they get removed from the colony. And so you need to avoid inbreeding by increasing the genetic diversity of not just the queen, but also of the source colonies where drones come from that would mate with those queens. The queen's quality is also affected by her mating conditions, in this case, the quality of the mates that she mates with. So not only does she need a sufficient number of drones to mate with, but you also have to pay attention to the natural mating conditions of the queens in terms of the availability of drones when the virgin queen is ready for mating. So are there enough drones that are sexually mature, which is very important during split time in the spring? Are there sexually mature drones, sufficient numbers of them in the fall during queen replacement? And so those are important questions that one needs to ask so that the queens can mate properly. We know that queens, Apis mellifera, depending on the subspecies, but on average they mate with between 12 and 14 drones, but potentially early in the spring, very early in the spring, or very late in the fall, they may not mate with that many drones because there are not enough sexually mature drones for them to mate with. And so you also have to ask what's the quality or the breed of the drones that those queens are mating with. So I'm going to tell you a little bit about drone biology that might inform us of why sometimes you do see drones in the reproductive season, but they're actually not quite ready for mating with the queen, and that might be problematic. So a colony produces thousands of drones during the swarming season only if the environmental conditions allow it. So if there's abundant forage and the colony is strong enough. So we've done a lot of work on what the minimum size of a colony must be in order for it to start investing in drone production. It's really expensive to produce drones. So typically small colonies, incipient colonies, first-year colonies, or colonies that started from swarms typically do not have the resources to produce drones. And so you can see that the production of drones coincides with the production of queens. This queen in this picture has already emerged. he might be still roaming around the colony, and that is kind of playing detective that this queen may not be very old, because typically workers will start chewing away the tip of these queen cells, and so this queen might still be a virgin around. And right here you see some drones emerging, coincide with the production of queens in nearby colonies. So something interesting about drone biology is that because drones only ejaculate once, if they're lucky enough to mate with a queen, all the sperm that they ever need will be formed before the drone emerges from his cell. And so what we call sexual maturity in drones is not that they need to generate sperm, but sexual maturity means the time that it takes for the sperm to migrate into the seminal vesicles of the queen before being able to inseminate her. The book says that it takes about 12 or more days for this to happen after the drone emerges. But our research has shown that it takes longer for these drones to typically be sexually mature, taking more than 16 to 18 days. In fact, some work that came out of Dr. Dave Tarpey's lab a couple of years ago, looking at drone quality, determined that drones are at their peak of sexual maturity at around 30 days post-emergence. So it's much longer and later than we thought previously, or that some of the old literature said. Sexually mature drones have about 10 million sperm cells in their seminal vesicles, or about a microliter of semen or 1 millimeter cube of semen. And so this is an inverted endophallus of a sexually mature drone, whereby you see the bulb at the end with the mucus that keeps the semen oxygenated for a while until the semen gets to pass the vaginal valve fold of the queen. But as we probably all know, a drone only provides a portion of the amount of sperm cells that he possesses into the queen's spermatheca. And it's actually believed that only 3 to 5% of a drone's ejaculate actually makes it up to the little duct that goes up to the spermatheca of the queen. That takes about 48 hours. So, and a well-filled spermatheca contains anywhere from 3 to 5 million sperm cells from all of her drone partners. This is another example of the bulb of a drone that has been exposed. And this is how we and others collect semen for artificial insemination or for assessment of semen quality. And so this information about how little sperm a drone contributes to a queen and how much sperm a queen needs for being properly mated also explains why she needs to mate with so many drones in order to have a full spermatheca upon mating. And so one of the work of the studies that we have done in my laboratory is looking at the effects of pesticide contamination, in particular beekeeper applied miticides that are kept in the wax matrix, its effects on drone reproductive quality. So for these experiments, we did 3 treatment groups and 1 control group, whereby we had plastic drone frames that have the drone cell pattern, so specifically for drone production, and we coated them with a very thin layer of pesticide-free wax, just almost like having wax foundation, but in this case of drone cells. And then we sprayed them with acetone that either was pure acetone for the control group that had no pesticides, or acetone that contained a combination of either the 2 previously most common miticides, fluvalinate and Coumaphos, or you might recognize them as Apistan and Checkmate Plus, a pyrethroid and an organophosphate at the field relevant concentrations that were found in a very important paper that I will talk about later. The second treatment contained chlorosalinate and chlorpyrifos, also at field relevant concentrations. So those were the top 2 agrochemicals that were found in wax samples in commercial beekeeping operations here in the US in that study by Mullen et al. And the last one is amethyst, the most currently used miticide to date. And so once we coated with equal numbers of sprays per frame for each treatment, all of these frames were placed into source colonies, strong source colonies, where we allowed them to draw out the comb and then for the queen to lay eggs either into a control frame or an experimental frame. So we allowed those frames to be drawn out and for the queens to lay eggs naturally. And then when we got something like this, where we had several drones being produced in that colony and they were about to emerge from their cells, we took those frames and placed them into incubators at 34 to 35 degrees Celsius. And every day we would check for the drones emergence. They were placed in the incubator with a frame with nurse bees and some food for them to feed. So every day we would collect the emerging drones. We would label them with a paint mark on their thorax, a specific color for each day. And then we placed them back into their source colonies. And we waited at least 16 days for the drones to reach sexual maturity before we collected them for sperm viability analysis. So we took for some 16, for some more than 18 days post-emergence. And for some, we wanted to see if they would still contain viable sperm at about 10 days post-emergence. And we collected them from the colonies with the paint marks, so we knew exactly how old they were. And then we would place them in what we call these bug dorms, these insect rearing cages. And then we would apply pressure to their abdomen like you do for instrumental insemination, and the endophallus would evert. And if the drone was sexually mature, they would express the pool of semen at the tip of the bulb. And then we collect the semen with a syringe, one syringe per individual. And then we would mix it in a diluent containing two dyes, Cyber 14, which dyes the acrosome or the head capsule of viable or alive sperm cells. That fluoresces is green, and propydium iodide, which attaches to the head capsule of inviable sperm cells, like shown here in this picture. And so this sperm cell is viable under the scope. The head capsule is fluorescing green, whereas these ones are fluorescing red because they're no longer alive. And something interesting to see is how long the tails of these sperm cells are compared to the size of the head for bee sperm. And we would use a Nexalom cell counter that actually does these calculations for us automatically now, instead of doing it by hand like I used to do when I was a postdoc. And so this gave us very troubling information. We found that we found significantly lower sperm viability in mature drones that were reared in the pesticide-laden wax compared to the control drones that were reared in the pesticide-free wax. So whereas the control drones had about an 80% sperm viability, the ones that were reared in the wax containing fluvalinate and coumaphos and the wax that contained chlorpyrifos and chlorothalonil had almost 40 to 50% less viable sperm. And the same pattern was seen in the drones that were reared in the amethyst-laden wax that had about a 20% drop in viability compared to the control frames that were in the same colony in pesticide-free drone foam. So the information that I gave you about drone availability during the spring and also the fall is important, but also the quality of the drones is important. So there's a lot of drones out there, especially near either agricultural sites or in areas where the bees are heavily treated against varroa, that may have a full load of sperm cells, but it's actually not viable sperm. So those are contributing to the queens not having high quality. The age and the vigor of the queen is also very important. So you have to keep track of your queen's age in order to know how old she is, of course, but also how much sperm she's used so far. So we know that the queen has this sperm storing organ, the spermatheca, what it looks like in unmated virgin queens, clear inside, whereas this is what it looks like after they've removed, of course, the tracheal nape that oxygenates the spermatheca and keeps the sperm alive. And so this picture, by the way, was taken by my friend and colleague, Sue Colby, whom you probably met before in other meetings. And so this study looked at the number of sperm in the spermatheca of queens or of mated queens that were either newly mated within a few months after mating, that were mated a year later, or that were alive a year later and were alive 2 years later. And as you can see, there's a dramatic decrease in the number of sperm, viable sperm available in 1-year-old queens compared to 0-year-old queens. So more than 50% of the sperm that a queen possesses is gone in her first year, and then it drops a little bit more, about 20 to 30% more in the next year. And so that is also, this is one of the reasons why we're losing queens prematurely, is because they may not be mating with drones that have fully viable sperm, and they're also running out of sperm prematurely, and that's why perhaps supersedure or queen replacement by workers is happening more than now than ever. Another interesting aspect of semen inside the queen spermathecae is the fact that the sperm in mated queens behaves differently and moves differently depending on how old the queen is and how long she's been keeping the sperm for. This is the sperm inside a previous or recently mated queen. The tails of that sperm move in circles. It's coiled up. You see that they're kind of tangled up in here in clumps because there's so many sperm cells and they have such long tails. Whereas, and so they move in circles and they move actually kind of fast. And I've seen this using a computer assisted sperm assessment machine that they use for stallion sperm. We've tested it with bees and I can actually see the sperm moving around this way. But a couple of years later, the sperm motility changes. The sperm uncoils as the queen ages, and as there is more space in the spermatheca, and they start moving. Instead of moving in circles, they start moving longitudinally, and they uncoil. As you can see, the length of the tail of the sperm cell fully uncoiled. So you can see the speed of queens drops dramatically, or the speed of the sperm inside queen spermathecae drops by about 10 to 20% the first year, but dramatically by about 40% by the second year. And so that is one of these things that that occurs to us all, senescence. And so these sperm are senescing. They probably have a lot more oxidative stress that is contributing to these queens losing their vigor because the sperm is not as potent anymore over time. So you need to keep track of your queen's age, as I said, just so that you know what kind of sperm availability and quality they might have. So do you have your queen labeled? Do you have records of that queen? So you should keep notes for all of your beekeeping endeavors, including that of your queen's birth and year, at least, of where you cut them. So for that, you can definitely try several ways of catching the queen if you're not very proficient, or you're new to beekeeping and you feel rightly so afraid of handling the queen because she's so precious and fragile. You might want to use some of these queen catching cages, or you can do it with your own fingers, but always using some sort of labeling mechanism with a marker or clipping very end of one of the wings. You of course do that after the queen has made it so that you don't preclude her from flying properly. So it's always good to keep a type of technique for labeling your queens with your own pattern. We know that there's this international queen labeling nomenclature. So for our 2021, this is the year of white queens. Very unlikely that you will find a queen that would have survived for more than 4 years. So that's why there's only 5 of these colors in our laboratory. We sometimes don't pay attention to the color. We sometimes use pink markers or purple markers, whatever we have at hand, but we know exactly what year that queen is because we have very good records. You could use also these plungers if you don't feel comfortable labeling the queen so that you can do so and know where your queens are coming from. So again, is it time to replace the queen once you know how old she is? You need to try to keep a very vigorous queen. And commercial beekeepers, at least here in the United States, because we have such heavy agricultural practices, are nowadays, it's not uncommon for them to prophylactically be replacing queens in their operations yearly nowadays, or maybe every other year at least. Another reason why you need to keep track of your queen's age is because the production of her mandibular pheromones changes over time, and they become less attractive over time. So the queen right colony functions as a cohesive unit, largely because of the queen mandibular gland chemicals, in particular the pheromones that are transmitted throughout the colony by queen worker contact. It's at least a blend of these 5 chemicals. 9-HCA has an enantiomer or a mirror image of itself, and that's why that adds up to 5. And it's produced again by the queen's mandibular glands that causes behavioral and physiological changes among colony members. And so the queen produces all of those mandibular gland pheromones, and then she has these little dots of molecules of QMP when she's auto-grooming herself. And then the queen attendants or the queen's retinue workers will pick up all of her queen's pheromones, and then will start grooming themselves. And so they will bring most of those chemicals toward their forelegs and antennae. They will do auto-grooming, and that way they will be either translocating some of those molecules internally for their own physiology, or they will be sharing them with other colony members through trophallaxis and contact. And so at any given time, these workers in the colony don't have to come in direct contact with the queen. They have to come in direct contact with enough queen pheromone to know that the queen is there and that the colony is functioning properly. A previous student in my laboratory, Liz Walsh, did a lot of studies on the effects of pesticide contamination in the wax matrix on the chemical composition of queen mandibular glands. And so this, I just wanted to show you this paper in Bee World shows how queen glands are dissected. In this little pouch right here is the actual gland. It's filled with an oily, yellowish pheromone that's what's basically produced by the queen and provided to the nurse workers to pass around the colony to learn about her status and her quality. So QMP of a good queen elicits a very robust retinue response, especially of the nurse H workers that take care of the queen. So if you put you excise or take out the contents of the queen mandibular gland and put it on a filter paper, you will get a very fast response of workers being just highly attracted to that smell. QNP also inhibits worker ovary development. We know that under certain circumstances, certain lines of workers will activate a few ovarials in their ovaries and allow them to lay fertilized eggs that turn into diploid into haploid, of course, drones. And this is a picture of activated ovaries in a worker compared to a virgin queen, compared to a fully mated queen. QMP also inhibits queen rearing by workers. So you always typically see these cups. Of course, this one is turned on its side, so you can see the cups inside, and this one actually has an egg inside that was laid by the queen. So you typically see these starter cells inside the colony, but they typically remain unoccupied unless there is either a very low titer of QMP because there's a lot of crowding in the colony, and that's what happens during swarming. That's one of the triggers for queen rearing prior to swarming, is there's a decrease in the production of, or a decrease per capita amount of QMP because there's so much crowding. Or if there is no queen, or the queen is failing and she doesn't smell right, then the workers start building queen cells. And that can happen in the case of supersedure. So when we're managing queens and learning about queen right colonies and or detecting where the queen might be, if you start seeing something like this in your colony with a bunch of queen cells in the middle of the queen brood nest area, this might be either a case of supersedure, which is the replacement of the old queen or not vigorous or low quality queen by the workers to create a new queen, and they choose a worker larva from the brood nest area, and they change the orientation of that cell, vertical now instead of horizontal, and they start feeding it, that larva, royal jelly. Or they can also, sometimes the queen will have already been gone because she was failing already and she's dead, and so that's what you would call emergency queen rearing. These queens from supersedure and emergency queen rearing are not always the best possible queens. Why? Because sometimes the workers don't wait, the workers wait until it's kind of already too late to choose which larvae they will feed royal jelly to. And the longer that the workers wait for that larva to be fed royal jelly, the lower the reproductive quality of that resulting queen. My work has shown that if the workers feed royal jelly to these larvae, either right after hatching, right after the egg hatches into a larva, so during the first larval day, compared to waiting 2 more days so that the larvae are 3-day-old larvae by the time they get their first meal of royal jelly, the overall productivity of that colony will drop by 30 to 40% in terms of amount of home built, worker production, food storage, etc. So it's a dramatic, it is of dramatic importance for us to know exactly if our queen is there at all times, knowing whether the queen is the one that we had purposely put in the colony. Of course, this is with a grain of salt. There's a lot of what people call natural beekeeping, in which they allow the bees to do whatever they really please with their own colonies when there is very little management. And so if that's the style that you choose, at least learning what to look for will probably help you avoid some issues associated with poor queen colonies. Okay, so there's other aspects of queens that are important to look at, especially pesticide exposure. I talked a little bit about pesticide exposure in drones and how it affects queens. We know, not surprisingly, as beekeepers, that honeybee colonies accumulate several environmental contaminants at levels that are detrimental to the health and productivity of the colony. And some of the work in terms of pesticide regulation uses honeybees as the kind of the landmark subject of study for pollinator health, and also kind of the canary in the coal mine in terms of looking for what pollutants and chemicals pollinators are being exposed to. And now more and more we see that the interaction between pesticides stress by varroa mites and disease are contributing to the decline of honeybee queens and honeybees colonies in general. In particular, because I'm interested in reproductive quality, we know from even the early 2000s that exposure to the miticides fluvalinate and coumaphos affect the size of the ovaries in queens, the viability of the sperm inside queen spermatici, and they decrease the mortality, survivorship, sorry, increase mortality of queens. These studies back then were done only with very high doses of these chemicals. They were placed topically on the queens. They were done one at a time. Whereas now we know that the wax matrix contains all of these chemicals at once because unfortunately there's not very good filtering systems that clean up the wax that we keep using over and over and over again that keep accumulating all these chemicals. So we know that beeswax is lipophilic and at least commercially available wax, and it's contaminated with several agrochemicals. So I had said at the beginning that we base a lot of our recent work, but it's completed now, but it was in the last 5 years. We looked at this study that was published by Mullen et al. In 2010. They followed about 250 commercial beekeeping operations in the United States, and they collected wax, bees, pollen, and nectar from these colonies. And so I'm showing you the top part of the table of the wax pesticide residues. And the first two agrochemicals were the insecticide chlorpyrifos, which has now been shown to cause fertility issues in men, human people that are pesticide applicators of this very harmful pesticide, and chlorothalonil, a fungicide that's used in agro ecosystems. But not surprisingly, the top two and then the fourth chemical or fifth chemical are the beekeeper applied fluvalinate, coumaphos, and metabolite of amitraz. So we wanted to see how those chemicals affect queen quality. So to do that, we reared queens in the way that queens are reared in mass, using plastic queen cups that are put into a what we call a queen rearing bar after grafting, and they look similar to a natural queen cell once the nurse bees have decided to start rearing those queens. So we coated each cup with about 200 milligrams of molten pesticide-free wax, certified pesticide-free wax. And then we used these three experimental groups similar to the work that we did with the drones. We did fluvalinate and coumaphos on its own because it was on its the top two chemicals, and they're no longer used, but they're still widely present in wax. Even today, because there's a paper that came out a couple of years ago by Trainer et al. That confirmed that still today, all of these chemicals are present in wax matrices around beekeeper operations. Then we used amitraz on its own as its own treatment group, and then the two agrochemicals at those field relevant concentrations that were found by Mullen et al. So we grafted 1-day-old larvae, and that is the physical transfer of 1-day-old worker larvae from the worker brood nest into those wax-coated plastic cups. Then we introduced those queen cells into queenless cell builders for the nurse bees to rear those queens. We allowed those queens to mature, and then once the cells were sealed, we placed each individual cell into a mating nuke and allowed those queens to emerge in their individual mating nukes and mate naturally. Once we confirmed that those queens had mated properly and they were laying worker destined eggs, we then used them for our experiments. And those mated queens were caged, placed into observation colonies that had 3 frames containing about 4,000 to 6,000 workers. And then we used them without knowing which colony was which or which colony had which queen. We took the observations. So we conducted choice tests and behavioral assays that are commonly done for this type of work. First, we collected emerging workers from a source colony. So we knew that they were emerging and less than 1 day old because we took a frame of emerging workers. We brushed off all the bees that were on that frame, and then we placed that frame into the incubator with a frame of food. And then at 24 hours later, we would come back to the incubator and we would collect all the bees that had emerged, the callow or very fuzzy, cute baby bees that don't really sting you. And then we place them in cages with a queen lure to allow them to grow up to the age of 5 days post-emergence. Meanwhile, we collected data from our observation colonies, and we did two types of observations. The retinue size, so the number of workers around the queen, and that's kind of like the picture I had shown you earlier. The QMP, queen mandibular pheromone, is highly attractive to workers depending on the quality of the queen and how that smell, how the bouquet of odors smell to the workers. Compared to this queen here, which doesn't have a very good, robust retinue, probably because she doesn't smell very good to, or strange, smells strangely to those workers. And so for the retinue size, we counted the number of workers that were around the queen in a 5-minute stretch. And we did that throughout the day for a minimum of 50 5-minute observations per queen. And we also did the same thing with egg laying, counting the number of eggs that the queen would lay in that period of time. Once those queens were done for, in terms of we had already collected the behavioral data, we sacrificed the queens, we dissected their mandibular glands, and we took the pairs of glands, one pair of glands, one of the glands from the two pairs. One gland was used for these choice tests that I will talk about. The other gland was done to do the chemical composition analysis of what the compounds were in those glands. I don't have time to show you the data for that, but basically we found that indeed the queens that were reared in the pesticide-laden wax had completely different chemical composition than control queens. And so therefore they smelled very differently and probably not as good as normal queens. And so we know that because then we did these cage bioassays where we placed those bees once they turned 5 days old in cohorts of about 30 workers per cage. And we smeared the content of the mandibular gland of either a control queen or an experimental queen in the same cage. And we counted the number of bees that were attracted to those contents of the mandibular glands. And indeed, we found the following. The number of bees on that slide was significant, on the pair of slides was significantly lower for the queens that were reared in the amethyst laden wax compared to the queens that were reared in the pesticide-free wax. Number of bees attending the slide or being attracted in that cage bioassay. And there was even a bigger dramatic difference in terms of lower number of bees attracted to the smear content of the mandibular gland in the queens that were reared in the fluvalinated chromophos group compared to the control group. We did not find a statistically significant difference in the number of bees attracted to the control versus the agrochemical laden wax queen glands. Something very interesting is that we found a significant drop in the number of eggs laid per hour in the colonies that were headed by all the treatment groups. So there were about 25 eggs laid per hour for these queens in the control group compared to less than 15 in the amethyst group and about a little over 15 for the other two groups. So there was a dramatic drop in the egg laying rate in the queens that were reared in the pesticide-laden wax. And the retinue size, the number of bees attracted to the queen around the retinue, was significantly lower in all of the treatment groups compared to the control queens. So this tells us also that the queens are not being treated um, as well as they should be for the control queens, because they were probably not smelling correctly to these workers. And so this has huge implications because they're not feeding them as much. They're not picking up as much pheromone. They're not attracted to that pheromone, which indicates that at some point they probably will get rid of that queen because she just does not smell properly the way that a normal queen should smell. So all of those things that I just showed you are some of the effects of pesticides on queen quality that we have to pay attention to. We know that queens are also exposed to both parasites and, of course, pathogens. They have, just like workers, they do suffer from parasitization by Varroa destructor, and not only from just the Varroa mites feeding on the queens, but also transmitting all of the some of the viruses that it vectors. In particular, the form-wing virus. When you see a queen like this, it's because your colony probably has a really, really bad Varroa case, and to the point that the queen herself was parasitized heavily by Varroa and emerged with the descriptive symptom of the form-wings. But they can also get viruses like black queen cell virus, in which the queen larvae, when infected, die and turn dark inside the cell. And then the workers have to remove this oily black queen that doesn't emerge because of the high viral loads of black queen cell virus. Queens can also get infected with the microsporidian gut pathogen Nosema, both Nosema cerana and Nosema apis, which we know causes dysentery and malnutrition in worker bees, but it also affects queen biology. So there have been some studies looking at the pathological effects of Nosema cerana on queen physiology, showing in particular that queens that have been artificially infected with Nosema cerana compared to uninfected queens have higher titers of certain genes, including vitellogenin and antioxidative genes. And these results tell us that the queen physiology is trying to overcome the pathogen load caused by Nosema by upregulating or higher or expressing at high degree these genes that, of course, will come at an expense to the queen's health. And interestingly, in terms of the composition of the queen mandibular pheromone, queens that are infected with Nosema cerana have a higher amount of 9-ODA, one of those QMP chemicals, a higher degree of levels of 9-HDA, another QMP chemical, and a significantly lower amount of HOB, another QMP chemical. And so again, the chemical composition of the overall bouquet of the QMP is significantly impacted by Nosema infection, which causes the workers to reject the smell of these queens, which have downstream effects in terms of queen care and productivity of the colony. We know that nutrition or lack thereof affects queen quality. In particular, nowadays there's a group that is now out of Dr. Julia Fine is now at the USDA lab at UC Davis, California, but she used to work with Dr. Gene Robinson, another one of the top 5 bee researchers here in the United States. And she developed this queen rearing cage device. It's a box that you should look into because it's I think it's an open access type of work where they're testing these laboratory system where they have the ability to put queens inside these little microcolonies and monitor their egg laying pattern really carefully. their plexiglass boxes. And so they tested that gadget that they were creating by seeing how feeding microcolonies different types of pollen supplements increased or decreased the amount of eggs that the queens laid. And so they tested the effects of using either fresh bee bread, frozen bee bread, or pollen patties on the egg laying capacity of the queens. And so this looks at the age of the workers, and here the number of eggs that the queens laid. And it shows basically over time that the queens laid significantly fewer eggs when they were fed a pollen patty that is just an artificial supplement made by beekeepers compared to the amount of eggs laid when they were fed either frozen or fresh bee bread. Even though they consumed more of the pollen patty, it might taste better to them. It's not as good for them in terms of the queen's productivity. So nutrition is also very important to the queen, not just the workers in the colony overall. I know that I am hopefully preaching to the choir when I talk about habitat fragmentation and how it affects queen quality. So we know that the loss of natural habitats threatens bee populations, and urbanization causes the loss of suitable bee habitats. In particular, I want to bring to your attention a study that was done out of North Carolina State a few years ago that looked at urbanization and how it changes pathogen and increases pathogen pressure on feral and managed honeybees. Just as a quick example out of that paper, basically shows that they tested how bees that were infected or subjected to paraquat or kind of an injection on their bodies with a little chemical that causes poor health, how just having either a low versus a high level of urbanization changes that individual's likelihood of survival. And so, not surprisingly, after they were injected with the chemical paraquat, the bees over time died about after a few days. But what's interesting is that the control bees had a much higher likelihood of survivorship over time when they came from an area of low urbanization compared to an area of high urbanization. And so urbanization increases pressures from pathogens and other ailments in honeybees, including the queens. We know also that the changing climate is affecting queen quality. Of course, severe drought and severe winters are causing the phenology of plants that are pollinated by honeybees to change, not just when they become available for foraging, but also the areas where they are available or stop from occurring because it's too hot or too cold compared to before. But the one study that I want to point to that Marla Spivak, one of my bee heroes, did many years ago in 1994 or so. She told me recently that no one had ever quoted this paper after I showed her this. She did it on seeing whether the development of queens would change, the developmental time of queens would change based on the temperature that they were reared in. And so she exposed queens at a kind of relatively normal temperature of 33 degrees Celsius compared to 3 degrees below 30 degrees Celsius or 35 degrees Celsius. And she tested this in both black line and yellow line queens. And what she noticed is that the developmental queen for the queens, as the temperature went up, the developmental time went down. And this is not necessarily good because then that means that the bees will have much less time to develop properly, and they probably will not be having the time to strengthen and have all the physiological processes before emerging as adults. They probably come out also much smaller in size compared to the queens that are reared in normal temperatures. And so we expect from climate change and warming temperatures that some patterns like this are probably already occurring in our queen populations, which could be another reason why our queens are not living as long. And finally, beekeeper practices, our own kind of, not fault, but our own practices are affecting our queens. So we need to be on the lookout for, as I said earlier, those supersedure cells. We need to also look at these drone laying queens. So if you find these frames like that have what looks at first like a normal worker egg laying pattern, but in the worker brood cell area, you see drone cells or drones being developed in worker cells with the kind of the dome capping indicative of drone production. That is showing the presence of a queen that has run out of sperm. She is a drone laying queen. She is still potentially unaware that she has not viable, inviable sperm left in her spermatheca, doing the whole process of fertilization of an egg, but it doesn't get fertilized until it turns into a drone, but inside worker cells. So what happens if you have to be always looking for the queen or for the presence of the queen? So you don't necessarily have to look for the queen, but you have to look for eggs and young larvae so that you know that you have a queen-right colony. If you don't find the queen and you don't see eggs, you have to start looking for these either supersedure or swarm cells in your colony, depending on the time of year, or this multiple eggs per cell that is indicative of laying workers. And so you want to make sure that you don't have a hopelessly queenless colony, which will increase the chances of having laying workers, as I showed before, and these workers will lay drone-destined eggs. And so you would say, well, at least that colony is producing some drones that could be fertilizing queens from other apiaries. But the problem is that the drones that are produced by laying worker colonies are not as good as queen-laid drones. So this study showed the drones from queen-right colonies versus laying worker colonies, and they looked at the weight of the drones, the volume of semen ejaculated by these drones, the number of sperm cells, the concentration of sperm cells in the volume, and the number of sperm cells per milligram of body mass in the drone. And all of these measures were significantly lower in the drones that came from laying worker colonies compared to the queen-right colonies. So it's not a good idea to just let your laying worker colonies start laying workers, laying drones for fertilization of queens. So this all tells us that you should probably requeen a colony every 1 or 2 years, because your young productive queen translates into populous colonies that will give you higher honey production. Those colonies that are headed by first year or early second year queens are less prone to swarming, and failed requeening attempts will hold colony buildup. In the spring, requeening failure will endanger your honey flow. So if you allow, for example, for the workers to do their own requeening, if it fails, then it will endanger your honey flow or having a large enough worker population to take care of that honey flow. In the fall, letting the workers requeen itself and failing will endanger your overwintering success of that colony, because it will be queenless. So the colony, the longer that colony is queenless, the harder it will be for it to be requeened later on, and you can have your colony turn into a hopelessly queenless colony with workers that will start laying eggs and becoming rebellious workers. So I hope that in this very long chat, if you've seen me talk, you know that I just don't stop and I go for it until I'm done. I hope that I've shown you some interesting and maybe unknown to you aspects of queen quality that need to be looked at so that we can have good queen management in our apiaries, starting from just the queen's own genetic background that she takes from her parents and also her drone partners, her vigor, how old she is, and the vigor that she may have in the spermathecae and the sperm therein, the aspects of pesticide and pathogens and parasites that are exposing our queens to all of these chemical treatments, the nutrition and how important it is, in particular, protein nutrition for the production of that really nutritious queen food. Of course, changing patterns of weather and fragmentation of our landscape, but in particular, our own beekeeping practices that need to have us as conscientious beekeepers that will always try to strive for having the best possible queens. So with that, I want to thank you for inviting me again to speak to your group. It has been a pleasure interacting with your board and John, Susie, Joe that invited me to give this talk again to you. I encourage you to go to our Facebook page. We have over 4,700 followers. We post all of the newest research that comes just not only out of our lab, but from important labs across the US and the world. And with that, I also want to thank our sponsor for allowing me to speak to your group today. And hopefully, I'm not sure if there will be any time for questions, but thank you again. And hopefully the next time you see me speak will be in person.
Oops. Done. Juliana, thank you very, very much indeed for that presentation. And indeed to all our participants. First thing I should say, I meant to sort of introduce myself at the beginning. My name is John Hill. I am chairman of Ulster Beekeepers, and I would certainly like to welcome all the participants who were with us this evening. We had from people from all over the planet, from Canada, from the USA, from all over Europe, including one from Cyprus, certainly from England, Scotland, Wales, Southern Ireland and Northern Ireland. So you've had quite an audience this evening, Juliana. And in fact, I think the questions are coming in fairly thick and fast. So I'm going to take a period of time, say maybe about 15 or 20 minutes. We will get through as many questions as we can in that time. Okay, so I'll start them off here. The very first question is from Declan. He says, do you clip your queens?
Good question. I don't, but Sue Kobe does. And she's my queen mentor. And when Sue Kobe comes to our laboratory, we do a lot of instrumental inseminations of queens. I wish I had time to tell you about this really cool study that we're doing now, looking at intragenomic conflict between Africanized and European queens. And for that, we need to inseminate the queens. And so once we inseminate them, we clip, or Sue clips them at the very tip of the forewing. It's just a little, very, very tiny clip that only you can detect. And that is in case the color mark comes off, which happens because the workers that groom the queen sometimes hate those colors and take it off. So clipping can be done, especially if it's after the queen has mated. You don't do that before she mates, it's after she mates. But it's not necessary. It's only kind of a double check that she is still the queen that you said she was. If you're not comfortable doing it, and or if you think, definitely if you're doing too much of it, then you can be perceived as injured by the workers and she can get superseded. So it has to be a very, very sliver that only you can detect that she's been clipped, just so that you know that that's the queen that you were referring to, especially for these studies that we do.
Okay, the next question is from Andrew. Did you do any testing of sperm viability with the organic acids? And if so, what did you find?
Great question. No, I haven't done that. long answer, short answer, there was no funding. I offered and requested that type of funding a couple of times, but the reason why it hasn't gained traction is that the organic acids don't stay in the wax matrix. They diffuse before they remain in the wax matrix. And so that's the argument against doing that type of work, because they don't remain in the wax. However, stay tuned, because we do want to do studies looking at just in general the effects of treating your colony with organic acids, especially oxalic acid. Not necessarily the availability or lack thereof of it in the wax matrix, but perhaps once you've treated colonies, how that might affect the queen's egg-laying pattern and or sperm viability.
Right, so the next question is from Norman. It's quite a long sentence. I think he said, so once we have a queen right colony producing honey, we should rear a new queen from her and develop this daughter on the side, either in a nook or apodia, and then at the end of the summer replace our queen with her daughter. So I think he's really just talking about bringing on a new, younger.
That is a great idea. So commercial beekeepers suffer more from queen failure because they are exerting their queens to degrees that we backyard beekeepers don't necessarily do. So requeening every year might be a little overstretched for a backyard beekeeper that doesn't use a lot of chemicals and is very conscientious and careful. But let's say you have a queen, she's still very vigorous the first year. You might not want to replace her that year, but definitely keep her so that in the early spring, as soon as you can rear queens from her, you rear queens from her. That's the way that you should do it, is locally grown and operated type of production of queens, and making sure that there are enough drone populations in your area. And again, remember that queens made about a kilometer or more away from their colonies to avoid inbreeding with relatives, so that they avoid that shot brood pattern. And so it's not enough for you to have drones of good quality and in high numbers in your area. You need to know if there are drones about a kilometer away from where your queen is produced that she will be mating with. But yeah, always try to be proactive, in rearing queens from your most productive colonies is the greatest thing you can do as a small queen producer.
Right, okay. I think actually the next question is really for me to answer, Juliana. It is from Connor, and he says, when can I see the recordings again? We're not making any arrangements about this at the moment, but in fact, when the series of webinars are over, which will be after the 18th of February, we will make an announcement on the Ulster Beekeepers website about that. So just watch this space, as they say. Our next question is from Seamus. He said, first, thank you for a very interesting, informative presentation. Could you perhaps comment on the difference between supersedure cells and swarm cells?
Yes, absolutely. Swarm cells you will find at the bottom if you're playing detective, because these queen management essentials are basically you playing detective of everything that can be going wrong or it's going right with your colony. So first is the timing of year. Swarming occurs typically in the late spring, probably in your area, right? In the UK, when do you get swarming, John? In May,
June.
Oh, yes, sorry, yes, just about
May, June, yeah.
time. April, May.
But for us, you guys are gonna laugh, but for us, it's gonna start in late February, early March. So anyway, so first is the time of year. So if it's the spring and you have a very populous colony, you're likely going to see more swarm cells than supersedure cells. And so you will see them at the bottom of your frames where there's the most space, at the bottom of the frames or on the edges where there's the most space. that's indicative that those cups were made for swarming. Supersede, and that's for colony fissioning. And so that's the colony is actually doing great, because if it wasn't, then it wouldn't have a large enough population to split into two. Supersedure cells occur in the middle of the brood nest area where the worker cells are. They're not at the bottom, and they are basically restructured worker cells that get changed to the shape of a queen cell in the center. And you would see them throughout that worker brood nest area. More likely, and so then for the supersedure ones, the question is the queen there or not? If you find eggs and or if you see the queen, that's a supersedure. If you don't see eggs and there's no queen, then it's emergency queen rearing. The queen is already dead. But those two latter cases occur in the middle of the brood nest area, not at the edges at the bottom.
Right. Okay. The next question is from Ervis. In addition to the impact of the waste that is in the wax, where they reduce the quality of the drone sperm, can a sick drone infect the queen in the mating flight? I think that means
the answer is yes. Yes, there is transmission. There are STDs in honeybees, and there has been work showing that drones can pass viruses to the queens during mating. Yes.
All right, okay.
And there's also been some work showing that there is vertical transmission of viruses from the queen to the eggs, to the offspring.
Okay.
So it's not just horizontal transmission during mating, but there's also vertical transmission from the queen to the offspring.
Right, okay. This one, David. Do you encourage your bees to make their own comb in preference to using foundation due to contaminants?
Yes and no. When we do studies looking at, of course, the effects of pesticides, we encourage the bees to produce their own wax. The problem is that it's very expensive to do so. And so one thing, expensive in terms of worker labor, their time and effort is spent on comb building. We like drones. I love drones because, you know, I work with reproduction, so I love seeing colonies that produce a lot of drones. Most beekeepers don't, and so they use foundation that is only for workers, and then they discourage drone production. In our colonies, we always have frames that only have a small strip of worker foundation at the top. So that gives the workers a template to start off with for worker production, but then it gives them the free space at the bottom so that they can rear as much drone comb as they want to. And so we, based on our work, we not only encourage you to take care of your queens and kind of see if they should be replaced every year or two, but we also encourage comb replacement every other year or so that your wax is not full of contaminants. But we understand that it's expensive in terms of comb production. If you don't use a lot of chemicals, if you do not have your bees on agricultural fields, then that can be a longer wait time. So what we suggest is not pulling all of your frames at once, but just doing the oldest ones first and having kind of a rotation paradigm so that then you only have a few frames at a time that are newly being drawn out by your bees.
Okay. Next one is from an anonymous. It says, does the USA use more pesticides in agriculture compared to the UK and Europe?
I don't know the answer to that. I should. I know that they have they use different kinds of chemicals, and because of just the size of the acreage that we have in the US, I would suspect that indeed we do use a lot more agrochemicals than they do in Europe. I don't know the regulations in Europe, but in terms of the US, there is no regulation or no such thing as organic honey, and that's because it is almost understood that it would be nearly impossible to have a radius of about 6 kilometers around a colony that would not have any kind of chemicals being used for any kind of crop production here. So yeah, especially with the soy belt and the corn belt and the all of those in the Midwest, there are tons and tons of chemicals being applied.
Yeah, I think I would tend to agree. There'd be very few places in the UK that could in fact produce organic honey because, because of chemicals. There might be certain areas in the Outer Hebrides or maybe in certain parts of Scotland and the Highlands, but very difficult. Anyway, the next question is very good talk. Is there any data on the effects of oxalic acid treatments on queen QMP?
No, there isn't. So that's one on my to-do list. I hope to have some more time now that I have 2 students finishing up their PhD this year, and so they're not occupying the colonies anymore. We have about 40 to 60 colonies in the research apiary every year, and my students are basically, they take over those colonies and use them for their research. But this year we have 2 students that are no longer using bees, so hopefully I'll get to work with some of these colonies for my own research interests, which include that very question about the oxalic acid.
Right. Okay. For Mick, it's due to the buildup of chemicals in wax over time. You mentioned it reduces fertility, egg laying rate, and AMP reduction. How do feral honeybees deal with this?
Well, for one, they're not exposed to the beekeeper applied chemicals, which are the ones that are found at highest prevalence. But the answer is, we don't know. We have a site. We have a site in South Texas, which is called the Welder Wildlife Foundation Biological Station, where over the last 20 years, my collaborators and I have, well, haven't been doing it for 20 years. I've been doing it for 8, but before my time, they were able to locate about 110 trees that have feral bees in them. And so we've been monitoring their, not just the Africanization process, but also their levels of disease. And this year we're going back to look at the levels of viruses in these feral colonies. The problem with these studies with feral bees, like if you follow Dr. Tom Seeley's work with feral bees, is that you can't really go into them to collect samples of the comb, and you can't really break them open and look at everything that's happening inside. So there's very little information about pesticide exposure of feral bees. I suspect that it's very minimal because they are in places where there's almost, at least the ones that we've studied, they're kind of in forested areas that are not very close to agricultural fields. But there could be areas where feral bees live that also are near agricultural settings, and we don't know. But all we know is that they are survivor stock because they're still there unmanaged for this long. So they have to be doing something right.
Mm-hmm. Well, we'll come down right to the last few questions, really, Juliana. There's one here from Andrew. It says, does synthetic QMP help avoid swarming?
They've tried using that, and some people have some success of doing increasing QMP to avoid swarming. But the reality is that, like every other sexually reproducing organism, honeybees are wanting to swarm because they are ready to reproduce, and it's that instinct that is very difficult to avoid. So swarm avoidance or preparations to contain swarming are are not very successful. The best possible way is probably to be proactive in splitting your colony before swarming occurs, because everything else you can do, for example, even plugging the entrance to prevent the swarm from escaping, is just a very temporary fix to an instinctive kind of behavior that is indicative of a bigger thing, that your colony is large enough and it needs to split because it's gained that ability to reproduce, and it's just instinctive.
Right, okay. There's a question here from Tim, and it's a question, in fact, that I probably would like to have asked as well. Do you have any recommendations for treatment for Varroa which avoid toxins affecting queen or drone development? For example, oxalic acid vaporization.
So I don't know your, John, maybe you can tell me what your regulations are for the use of oxalic acid in the UK. But here we have three authorized uses: the oxalic acid sublimation with the crystals, the dribble, and the spraying method. In our apiary, the most successful Varroa control method that we've had is with the sublimation of crystals. It has been very effective. But we do, because we have, we do a lot of research with Varroa, and so we have very heavily parasitized colonies. To try to keep them from collapsing before the fall, we use Apivar as well, so Amitraz. So, but whatever you do, you should rotate your treatment, potentially using oxalic acid in one time of year, and then if you're doing 2 treatments, maybe using formic acid the next time, for the second time in the year. Always rotating your treatment effect. And you can use the biocontrol methods or the mechanical methods for IPM. You can do the stopping of the brood cycle, the colony cycle, by caging the queen for a couple of weeks to prevent any brood from growing, and therefore stopping the reproductive phase of Varroa, and or culling all of your drone comb. So putting in drone frames, waiting for those drones to be capped, and then taking them out of into the freezer, making sure that you don't forget about those drone cells so that they don't emerge. The drones don't emerge in your colony, because then you would have what they call a Varroa bomb full of Varroa that came out of your drone cells.
Certainly, just for your information, certainly oxalic acid is used here both by dribbling and by sublimation. We also do have max strips, which is then is formic acid, and we do have Amitraz. The one thing we do, and thymol, of course, is the other one. The thing we do not have is Coumaphos, which is not legalized in the UK.
Yeah, and I guess these studies that looked at Varroa resistance to both fluvalinate and Coumaphos were done here in the US. And unfortunately, there was a paper that came out last year where they've reported some populations of Varroa now being resistant to Amitraz, which is problematic, because it's the only treatment that beekeepers, at least commercial beekeepers, are using nowadays, because they're not relying on the organic acids, because they are so temperature dependent, and so they don't want to take any chances with treating thousands of colonies and seeing what happens. They need to treat kind of prophylactically, which is, I think, also one of our bigger problems, is that then the majority of our wax that comes from commercial operations is heavily contaminated, because they treat colonies maybe 3 or 4 times a year prophylactically to avoid collapse due to varroa.
Yeah. Well, I'd like to thank everybody for bringing these questions. And as I say, we'll just have one more, but those who have asked questions, I'm afraid, unfortunately, our time has run out. But the last question really is from Daniel, and he said, what is the optimal temperature for development of a good queen in an incubator?
That's a good question. We keep our queens in the incubator at approximately the same temperature that you keep the brood nest area. So anywhere from 33 to 35 degrees. And as you just saw from Marla Spivak's paper, the warmer the temperature, the shorter the developmental time. So definitely do not go over 34 degrees Celsius. Somewhere even slightly cooler might be a better ticket to delay the development and allow these queens to be have enough time to develop. So let's say 33 degrees as the median.
Right. Well, Juliana, thank you so much for delivering what's a quite excellent lecture, and the comments that are coming back to us here are all brilliant. Most people coming through are leaving comments that really were very impressed by this talk.